welcome back to your A-level biology teleclasses. This is Photosynthesis from CHSA. Today we're going to be starting with your grade 12 bio content, and since it's the first lesson of grade 12, I'll start by talking about the papers that you study for in your grade 12, and then we'll look at our topic for today, which is going to be an introduction to photosynthesis. Before we start, please make sure you are comfortably seated and you have your notebook and a pen so that you can take notes as is necessary. So let's get started. As you know, there are a total of 6 papers that you sit for in your A-level biology. 3 of them you can sit at the end of your grade 11, and there are 3 more that you sit at the end of grade 12. And at each of these levels, there are 2 theory-based papers and 1 practical-based paper. In your AS, Units 1 and 2 were the theory-based papers, and Unit 3 was the practical-based paper. Similarly, in grade 12, Units 4 and 5 are going to be your theory-based papers, and Unit 6 is going to be the practical-based paper. If you look at the duration and the marks for them, you'd see that these theory papers now are slightly longer and therefore a slightly higher mark as well. So make sure you keep that in mind as you do questions and you practice these papers. Another thing to remember is, since you're doing the new IAL syllabus, that is IAL Biology from 2018, your unit codes are slightly different from the ones that you would have in your current past paper booklets. Okay, for example, Unit 4, you're sitting for the WBI 1401 paper, which would not be what you would have in the past paper booklet. And as you can imagine from that, that means there are definitely some changes to the content itself, and but for the most part it's the same. There are some changes to the content, so you can still practice the old papers because we don't have many reference papers of the new syllabus, so you can use the old papers for practice. But the changes, if you want to know for yourself, you can go to the Edexcel website, which I've given at the bottom here, and you can look up the materials that they provided there too. So make sure though you're looking at the Biology 2018, not the earlier versions, because those are still available too on the website. So let's start with topic Unit 4 because we're starting with that. So like any other unit, theory unit, Unit 4 also has 2 main topics to them. So we have topic 5 and topic 6 in Unit 4. And in topic 5, as the name suggests, you can tell what are some of the content that you'll be learning here. And since our first lesson is about photosynthesis, and photosynthesis is where energy capture starts, that's what we will start in with too. So in this topic, you learn about how the energy is captured in photosynthesis and how the organisms can feed on those plants, and those organisms will be fed on, leading to this energy flow within this ecosystem, which will then be followed by the carbon cycle. So you learn about the carbon cycle and how changes to the carbon cycle can lead to global climate change. And you would also be learning about how populations can change over time as well as a result of natural selection and mutation, which can potentially lead to speciation as well. So these are some of the major topics. There are a few others which I've not quite covered here, but you will know as we go through. When we go to topic 6, we talk about the content that is there too. So starting with photosynthesis. I'm sure you've learned quite a lot about it in your O-levels, and there were some relevant materials in your AS as well. So what we'll be doing in today's class is we're going to look at, basically recall the things that you've learned in your O-levels about photosynthesis, and then bring the things that you've learned in your AS relevant to this as well, so that we can combine the two together, so that that knowledge can then be used for our future lessons. Okay, so for the most part it's going to be just recalling the things that you know, but there are some new content in this, so make sure you have a look out for those as well. So photosynthesis, what do you know about it? Well, for one, you would know it's a process that occurs in plants, and it's the process where plants will capture the energy in the light so that they can synthesize sugars for themselves. And you would have also learned about the overall reaction for it, where you have carbon dioxide and water in the presence of light energy with the help of chlorophyll forming glucose and oxygen, right? And since this is the overall reaction, you would know that this is not all of the things involved in it. This is just the summary of it. So at your A levels, you learn about what other components are involved in this reaction, and mostly about the details of this process. If you look at the chemical equation for this, you would know that to make one molecule of glucose itself, you would need 6 molecules of carbon dioxide and 6 molecules of water, and it's also going to produce 6 molecules of oxygen. So how would then the plants use these reactants? How would they get these reactants for photosynthesis to occur, and how would they use the products of this? Well, where do you find the carbon dioxide? Carbon dioxide would be definitely found in the atmosphere, so that's where the plants get them, and it's going to enter into the plant through the stomata found on the underside of the leaves. So they diffuse in through the stomata into the leaves so that photosynthesis can occur, and the water, mostly found in the soil, will be taken up by the roots, and they're going to be transported to those leaves through xylem vessels that would be found within the plant. And then in the leaves, where you'd find this chlorophyll, the glucose is going to be synthesized. And the glucose, how do they use it? Well, for one thing, glucose is going to be used for respiration. In addition to that, glucose is going to be the initial molecule that will then be converted to form those other organic molecules that the plant needs as well. You would have learned quite a few of those different molecules in your AS levels, like the carbohydrate structures, proteins, nucleic acids, lipids, and all that. So all of those different organic molecules are basically made from modifications done to these sugars that are synthesized during photosynthesis. Looking at oxygen, how do they use it? Well, for respiration to occur, in addition to the glucose, they would also need the oxygen. So they use it for their respiration, and whatever is left of it will be diffusing out, and that will then be used by other non-photosynthetic organisms like you and me and all of the other respiring organisms too. And you would have learned that for this entire process to occur, plants have lots of different adaptations to them as well. So let's look at a few of those. We'll start by looking at the leaf, because that's the place where photosynthesis occurs. And you would remember, and you can see by yourself as well, the leaves themselves, they are almost always fairly thin, and they're very broad as well. So how would this be helpful? Well, for starters, having those thin broad leaves ensures that, so when it's thin, ensures that the light doesn't have to pass through many layers, and it ensures that you don't have a large diffusion distance for the carbon dioxide to enter and move through as well. Another thing is, them being broad ensures that there's a large surface area for the light to be captured as well. In addition to that, you'd notice that leaves always have this large network of veins in them as well. So those veins will be made up of different vessels, such as the xylem vessels and the phloem vessels, right? And those network of veins, in addition to providing the water that is needed for photosynthesis, also ensures that the lignified xylem vessels hold the leaf flat so that the light can strike them too. Okay, if we look up close to the cross section of the leaf, so this diagram too you would be familiar with. At each of these levels, they are adapted for photosynthesis to occur too. So for example, you would know that the leaf has many different photosynthesizing cells, like the palisade mesophyll cells and the spongy mesophyll cells, as well as the guard cells that make up the opening of the stomata too. And most of those chloroplasts would be found in these palisade mesophyll cells, which are found at the upper part of the leaf. So they are found close to the surface of the leaves, and they are very closely packed to ensure that just as light strikes, they can be absorbed. Most of that light can be absorbed by these palisade mesophyll cells. Another thing is, you remember that the epidermal layer here is going to be transparent, so it doesn't have any pigments in it, which ensures that any light that strikes will be passed into the photosynthesizing cells beneath it. And then looking at these spongy mesophyll cells, you know that they are irregularly shaped. So having a irregular shape, for one thing, ensures that there's a large surface area for gases to be taken in. Another thing is, it also allows for these small spaces, these air spaces, to be present. So those air spaces allows for a way by which the gases can actually move between these cells to the cells beyond as well. And if you look at the vascular bundle, what would be the function of it? So I told you already that for one thing, it holds the leaf flat because of the lignified cells. But in addition to that, the xylem vessels bring the water to the cells for photosynthesis to occur. It also carries with it the ions that would be needed to synthesize other molecules as well. And when photosynthesis occurs, the sugars that would be used, the sugars that are being formed, can then be transported through the phloem cells to other parts of the plant where they can be stored as well. And lastly, if you look at any leaf, you would find lots and lots of stomata on the underside of the leaf as well, and this allows for a way in which gases can be exchanged within the leaf and the atmosphere too. Now let's look at just one of these mesophyll cells. So in any cell, in plant cells, you would notice that the chloroplast, they always found at the periphery of the cell. So what would be the advantage of this? Well, if they are found at the edges, at the periphery of the cell, because of the large vacuole inside, it means that the water and the carbon dioxide doesn't have to travel too far into the cell for them to reach the chloroplast where photosynthesis can occur. Alright, so let's look at the chloroplast itself. You've learned about this structure in quite a lot of detail, so I'll just brief you about the major parts of it. So to start with, chloroplast, is it a membrane bound organelle or not? Well, you know it's a membrane bound organelle, and you know that it has a double membrane around it, right? So you have this outer membrane and an inner membrane making up the chloroplast envelope, and this envelope, these two membranes, ensure that the fluid that is found within the chloroplast is separated from that of the fluid outside. So inside we have the fluid filling that would be the stroma, and you know that on the outside of the chloroplast you would have the cytoplasm, right? And in this stroma you will have lots of different enzymes, lots of different things involved in photosynthesis to occur, and having this membrane ensures that only certain molecules can move in and out of the chloroplast. But in addition to that, it provides this compartment, these separate spaces for photosynthesis to occur, separate from those reactions that will be happening in the cytoplasm. Okay, and in this stroma, in addition to those different proteins and enzymes and all that, you have this huge network of membranes which are referred to as thylakoids. So a thylakoid is really this membranous structure that is flat. So you have these flattened discs that would be found within the stroma, and those will have a space inside, so that's called the thylakoid space, and the membrane that surrounds them together, that's called a thylakoid. And you would remember, so you can see from here too, we have these thylakoids sometimes arranged in stacks, and you have these structures connecting them two together. So whenever you have thylakoids arranged in stacks on top of the other, those are called grana. So I have one grana shown here, one granum here, another granum here, and another granum here. Altogether 3 granum are shown that in this particular diagram. So the term granum is for one, grana is for many. So there are 3 grana in this picture shown. And then what do you call then these structures that are connecting these different grana together? Well, there are quite a few different names, so I'm going to give you a couple of them. For the most part, what is used is lamellae. So they are called lamellae, that is holding that are connecting these different grana together. In your textbook as well, the term lamellae is used, but if you refer to any of the others, sometimes you would see that they are called stromal thylakoids, because these are basically thylakoids found within the stroma. Sometimes they are called intergranal lamellae as well, because within the grana, stromal lamellae again for the same reason. So any of these, we're talking about the same thing still. Okay, now let's look at the membrane of this thylakoid. So the thylakoid lumen, the thylakoid membrane, like any other membrane, is made up of a phospholipid bilayer. So you'd have a bilayer of phospholipids, and within this bilayer there would be many different proteins embedded. We'll look at some of these proteins in more detail when we look at photosynthesis in detail later. But you can have different carrier proteins, you can have different channel proteins, and you would have different protein complexes in them as well. And it is in one of those protein complex where you'd find the chlorophyll molecule embedded. So where do you find the chlorophyll molecule? Well, it's in a protein complex called as a photosystem, which we'll look at later. So it's found in a protein complex found in the thylakoid membrane, found within a chloroplast, which will be found in photosynthesizing cells, which are found in leaves on plants. So quite long. So if you want to know exactly where it is, that's where it is. Anyway, moving on. So we know that there are lots of proteins within the membrane and within the stroma here too, and some of those proteins actually coded by the DNA that would be found within the chloroplast. So this chloroplast DNA codes for different proteins, and you know that for the information, the genes on the DNA to be coded, to be translated to these proteins, you also need other structures, and those are going to be ribosomes. And the ribosomes that are found within the chloroplast, those are called 70S ribosomes. Okay, so these are the major structures, and with all of them you will have photosynthesis occurring, forming the sugars, and some of those sugars, if there's excess of it, they can actually be stored within the chloroplast by starch granules as well. So in some pictures of chloroplast you'll find these starch granules, in some you may not as well. But for the most part in plants, the starch are not stored in chloroplast, they're stored in other organelles called amyloplast, which are found in most of the storage organs. Okay, there would definitely be other ions and other structures that can be found in the chloroplast, but because most of them don't help identify the chloroplast from that of any other structure, I've not discussed about those. Let's look at another picture of a chloroplast here. So here we have an image, slightly three-dimensional image of a chloroplast showing the outer membrane and the inner membrane, and this here shows an electron microscope image of a chloroplast. As you can see, we have lots and lots of thylakoids arranged in stacks called those grana. So in the picture I showed you, there were just 3 grana, but in actuality you can have loads of them, right? And these would be the starch granules showed in black in this particular picture, and then those lamellae that are connecting different thylakoids together as well. So if you remember from before, this would be an electron micrograph which shows a higher magnification than the normal chloroplast that you can see in this light microscope image here. So with the higher magnification, it allows for higher, and the higher resolution allows for a more clear image to be seen. So we look at this and the entire process of photosynthesis in another class. With that, we're done for today's class. In our next session, we're going to look at the structure of chlorophyll, where it's found, how it's used by the plants, and how it can be isolated and identified as well. So till we meet again, please be safe, take care of yourself and your loved ones, and I hope you have a good day. Assalamu alaikum.